Hello everyone. In this video we are going to discuss about what's going on within a neuron when the neuron is actually doing nothing, or when the neuron is at rest. So, and hopefully in the next video we're going to discuss on how a neuron actually transfers its signal. So let's begin. Just for a quick review, let's just draw a rough sketch of a neuron so to refresh our memory. So this is the soma, these are the dendrites, and this is the axon. And here you have some terminals, the axon terminal. So this is the axon, this is the terminal. So a neuron which is not doing anything, or which is at rest, has potential energy in it, and that potential energy, well, is used to transfer signals from one part of the body to another, or from one neuron to another, or from one neuron to a muscle cell or somewhere else. So this neuron has some potential energy, and it can be used to transfer signals. So if I were to just focus or zoom here in the neuron right here, just maybe this part of it, and just zoom it out, just zoom in, I would notice that this potential energy exists due to a reason. And what is that reason? Well, the reason is due to an not equal, or due to an unequal distribution of charges. So what do I mean by that? Well, I mean that the outside is more positive, and the inside is negative. So this unequal charge distribution causes this potential energy. And if I were to measure it, if I were to measure the strength of this potential energy, or in other words, if I were to measure the voltage, which is the difference between the difference between electropotential, the electric potential from one part to another, or the electropotential difference, if I were to measure it, I would find that the voltage of a resting membrane, of a resting neuron, is actually minus 70 millivolts. And how do I get it? Well, what the scientists did was they actually put two microelectrodes. They actually put two microelectrodes, one in the inside of the cell, and they connected it to a voltmeter, and one to the outside of the cell. And the difference in the electrical potential energy from the inside toward the outside came minus 70, which is the voltage, or in other words, the strength. So, and actually the resting membrane maintains this potential energy. And how do they do that? Well, there are basically a couple of reasons for how they do that. Partly because of a pump called the sodium potassium pump. Well, partly because of this, but mainly there's a reason of channels which allow this to happen. Well, that's the best word I could use here. Of certain channels that allow this to happen. So first of all, let's discuss about how does this pump work? How does the sodium potassium pump work? And this is a horrible drawing I drew right here. So let's go down a little bit. Oops. That's fine. So let's discuss about the sodium potassium pump. So if I were to draw again a axon, and if I were to draw again the axon and just draw a pump called draw a sodium potassium pump, I would draw something like this. Well, there's a well, not like this. Let's draw it something like this, and like this. So So this is a rough sketch of the sodium potassium pump I drew here. So what happens is that outside the outside of the axon contains a large amount of sodium, ions, and really less amount of potassium ions. So you have more sodium outside and less potassium inside. And at the bottom, and this is the outside of the cell, outside, and the inside, towards the inside, you have more potassium and less sodium. So when you try to figure out the voltage, because there are a lot more sodium than there are potassiums inside, it creates a potential difference of minus 70 volts. So now let's try to figure out what's going on here. So you have some potassium, some sodium ions here, and this sodium, what the sodium potassium pump does is that when an ATP molecule comes along and it gives one phosphate to this molecule, and it gives a phosphate molecule, it gives a phosphate to this molecule and gives a lot of energy. That energy is used to take 3 sodium ions, so 1, 2, and 3. So it uses this energy given by the ATP to take 3 sodium ions and to push them towards the outside of the cell. And when it does, the shape would become something like this: 1, 2, 3, and you have this one. So you have 2 places here, and should draw it better. So you have, so the shape changes, so it opens, and when this potassium ion, when this phosphate group leaves, it gets 2 of these potassiums and joins them here, and it pushes 2 of the potassiums towards the inside. So you have, basically you're moving 3 sodiums outside and 2 potassiums inside. So this is what the sodium potassium pump does, and this is not quite, but this is partly the reason why this membrane potential is minus 70 degrees. Oh, not minus 70 degrees, minus 70 millivolts. Sorry. So this is what the sodium potassium pump does. Now, what is the main reason why this membrane keeps its potential energy the same, or keeps its voltage, keeps the strength of this potential energy till minus 70 volts? Well, the other reason is that if you look at an axon again, the axon has channels of proteins, made of proteins, and as I said before, there are a lot more sodiums outside, and basically this the sodium potassium pump doesn't really affect that much, but it has slightly effect, but it's not that effective of keeping that potential to minus 70. What the main reason is, which we are going to discuss right now, is, so you see there are a lot more sodiums outside than there are inside, and you have fewer potassium, right? And you have a lot more potassium inside, then you have fewer sodiums inside. So there's a difference. Now these channels of proteins right here are, well, they are more permeable. Okay, they're permeable to what? Well, they're more permeable to potassium rather than sodium. Now if you notice, there are a lot more potassium inside rather than outside, so potassiums would eventually want to go out because there are a lot more potassiums, right? And since the membrane is permeable, they can do it really easily. So potassium has a lot more chances of going outside of the membrane rather than for the sodium, which is sodium, which the membrane is impermeable to, cannot go in. So potassium has a higher chance of going outside because it's more permeable, rather than for sodium to get inside, which is impermeable to the membrane. So this is basically the reason why there's a potential difference. There's a potential difference of minus 70 volts. And other reason is that the inside of a cell, they also have certain proteins which are negatively charged. So this adds up to why we have minus 70 millivolts. And there are two terminologies right here which I typed it. So resting potential and membrane potential. Well, there is a word called the membrane potential, and membrane potential is basically the potential the potential energy of the membrane of a neuron. So if somebody says membrane potential, it's just the potential energy of a membrane of a neuron. And when a neuron is addressed, at that time the difference between electrical charges and the difference at that time when the neuron is addressed, at that time the potential energy is said to be resting potential. So, and the resting potential of a resting neuron is, well, minus 70 millivolts. This is the electropotential difference across the neuron. So this is how neuron is when it is not doing anything, or is at rest, or when it is not stimulated. It has a potential difference of minus 70 millivolts, which is called resting potential. And the potential energy of a membrane is called membrane potential. So hope you guys found that useful, and I'll see you in the next video. Thank you.